Good morning, my name is Kathryn from the Department of Biology here at BU, and a huge thank you to the CTSI for allowing me to present my research to you all, which has been focused on investigating the strategies for the rescue of cellular and behavioral deficits in a Nexmif-dependent mouse model of autism spectrum disorder. Autism spectrum disorders, or ASDs, are characterized by impaired social interactions, diminished communication, and repetitive behaviors. Nexmif is an X-linked gene whose loss is associated with the development of ASD and intellectual disability in both human males and females. At the functional level, Nexmif may play a role in the transcriptional regulation of genes implicated in early brain development. To study Nexmif ASD, our lab generated the Nexmif knockout male mouse model, as well as the heterozygous female mouse model, which display deficits in neuron morphology, dysregulated gene expression, ASD and anxiety-like behaviors, hyperactivity, and impaired memory. So given the presence of these impaired phenotypes in our mouse model, we wondered whether they could be effectively rescued, and if so, how and to what extent. So moving forward, my talk will be divided into two parts. In part one, I'll share with you how we've been using the strategy of postnatal gene reintroduction to rescue the knockout male mice. So given that the male mice have complete loss of Nexmif, we generated a Nexmif lentivirus containing a human Nexmif transgene and injected the virus bilaterally into the ventricles of newborn pups. And then during the adolescent period, we performed a series of behavioral tests to assess for the presence of ASD-like behaviors and comorbidities. And then at the end of each time point, we collected the brain tissue for biochemical analyses. So these injections were performed in 3 groups of mice: wild-type and knockout mice with a control lentivirus, and knockout mice with the Nexmif lentivirus as the rescue group. So indeed, we were able to confirm in the brain tissue that Nexmif was very nicely restored in the cortex and hippocampus of the rescue mice. In addition to the rescue of several synaptic proteins that are crucial in learning and memory function, such as GluA1, SYNDIG1, and TREC-B. We also found that Nexmif restoration could rescue dendritic spine density, which is a crucial part of neuron morphology in both the medial prefrontal cortex and in the hippocampus. However, we failed to see rescue in the dentate gyrus, which we believe could be due to a lack of viral penetration to that area. Now, at the behavioral level, we used the novel object recognition test to assess deficits in short-term memory. Indeed, we found that the knockout control mice showed impaired discrimination indices relative to the wild-type control mice. However, this was nicely rescued by Nexmif restoration in the knockout mice. Then we used the 3-chamber social test to assess for deficits in social behavior. So during the sociability test, we examined the preference of a test mouse for a stranger mouse versus an empty cage. Indeed, we see that the knockout control mice show reduced sociability, and this is partially rescued by the presence of Nexmif restoration. Then during the social novelty test, in which we test the preference of a test mouse for a familiar mouse versus a novel mouse, we saw stark reduction in social novelty preference indices, which was very nicely rescued by the presence of Nexmif restoration. We also examined grooming and anxiety-like behavior. In the knockout control animals, they showed increased repetitive over-grooming behavior, but this was nicely reduced by Nexmif restoration. We also used the elevated zero maze to examine anxiety by quantifying the amount of time the mice spend hiding in the closed arms. As we see here, the knockout control mice spent the majority of their time hiding in the closed arms, but this was partially rescued by Nexmif restoration in the knockout mice. Then we use RNA sequencing to examine gene expression changes in the knockout plus control mice. Indeed, we found significant dysregulation of gene expression in the knockout plus control mice relative to the wild-type plus control mice. However, this was nicely rescued in the knockout plus Nexmif mice. Lastly, loss of Nexmif is associated with impaired dendritic outgrowth, and so we wanted to see whether genes downstream of Nexmif regulation could rescue this specific phenotype. Indeed, our RNA sequencing dataset showed us that Philip1 is a gene that it was significantly reduced at both the mRNA and protein levels in knockout plus control mice, and we found that Philip1 is associated with stimulation of neurite outgrowth. So we wanted to see whether Philip1 restoration in Nexmif knockdown cells could rescue this dendritic growth phenotype. And so we knocked down Nexmif virally in cultured rat cortical neurons and then transfected in Philip1 to examine its effects on dendritic growth. Indeed, using shoal analysis, we were able to show that Philip 1 restoration could rescue the number of dendritic crossings, as well as the number of dendrites, total dendritic length, and the mean length. So to summarize the first part of my talk, we postnatally restored Nexmif expression in Nexmif knockout mice, which rescued several neuronal and behavioral deficits by adolescent ages. In cultured Nexmif knockdown rat neurons, restoration of the downstream gene Philip1 nicely rescued dendritic outgrowth and branching. So overall, we believe that these findings support further investigation of the therapeutic potential of gene reintroduction techniques for Nexmif-dependent ASD in human males. For part 2 of my talk, I'll share with you how we've been using the strategy of X chromosome reactivation to specifically rescue our Nexmif heterozygous female mice. So firstly, X chromosome inactivation is a process that occurs only in female cells, in which one X chromosome is chosen to be randomly inactivated, and this happens in order to balance the X-linked gene dosage between males and females. So as you can imagine, this begs the question for what happens in an X-linked gene when it has a mutation on it. Indeed, if we use our het female mice as an example, in which one X chromosome contains the wild-type functional copy of Nexmif, and the other X chromosome contains the mutated copy, this actually leads to the production of two cell types, Nexmif-positive cells and Nexmif-negative cells. These Nexmif-negative cells actually function similarly to the neurons of knockout male mice. And so in an attempt to reactivate this functional wild-type copy of Nexmif from the inactivated X in Nexmif-negative cells of the het mice, we came up with two strategies, the first being to pharmacologically inhibit facilitators of XCI. So to test this first strategy, we cultured neurons from het mice, wild-type female mice, and wild-type male mice as negative controls, and then we treated the cultured neurons for a total of 9 days with the various inhibitors, and then we collected the lysates for mRNA expression, specifically of Nexmif, and two other X-linked genes as controls, ATRX and MECP2. And indeed, we found that the inhibitor of DNA methylation, azopus resveratrol in the green bar, could very nicely increase Nexmif mRNA expression in the het female and wild-type female cultured neurons with little to no change in wild-type males and with no change in MECP2. And then we tested azopus resveratrol in vivo by using a unilateral brain injection method of injecting vehicle into one side and azopus resveratrol into the other side at the neonatal age of P1, and then collecting the brain tissue 14 days later for mRNA and protein quantification. Indeed, we found that azopus resveratrol was very nicely able to increase Nexmif mRNA in wild-type females and in het females at both the mRNA and protein levels, but it did have an effect on MECP2 and ATRX, indicating that azopus resveratrol has a non-specific effect. At a more translational level, we also wanted to see the effects of azopus resveratrol on human female forebrain neurons derived from iPSCs. So indeed, we found that azopus resveratrol was able to nicely increase human Nexmif mRNA, as well as at the protein level. However, again, we see this non-specific increase in human ATRX, indicating non-specificity of azopus resveratrol. So for our second strategy, we employed the use of the CRISPR-Cas9 system, or CRISPR-A, to specifically achieve increases in Nexmif expression. And indeed, we found that CRISPR-A was able to specifically increase Nexmif expression in hex cells at the mRNA level and protein, as well as in our human female forebrain neurons, with little to no effect on MECP2 or ATRX, indicating specificity. So to examine the effect of CRISPR-A in vivo, we generated a lentivirus and then injected one group of wild-type female mice bilaterally with the control lentivirus and another group with the CRISPR-A lentivirus at P1, and then 29 days later collected the brain tissue for mRNA and protein quantification. And without fail, we saw a very nice and specific increase in Nexmif mRNA and protein in the wild-type female mice, with little to no effects on MECP2 and ATRX, indicating strong specificity of this CRISPR-A system. So to summarize the second part of my talk, we found that in wild-type and het female mice, the DNA methylation inhibitor azopus resveratrol increased Nexmif expression but lacked specificity. However, specific activation of Nexmif by the CRISPR-A system increased Nexmif in hex cells and human forebrain neurons, as well as in vivo in wild-type female mice. And so overall, we believe that these findings support further investigation of the therapeutic potential of gene reactivation techniques for Nexmif-dependent ASD in human females. So as for future directions, for the knockout male project, we will determine the latest time point at which Nexmif restoration can still elicit neuronal and behavioral rescue in the mice. And for the het female project, we will generate X-linked TD tomato het female mice, which will allow us to distinguish between the wild type and knockout cells. And then we will use this fluorescent model to test the CRISPR-A virus to examine rescue at the neuronal and behavioral level in the het mice. And for acknowledgements, I'd like to thank my lab, the biology department, my NIH funding sources, and here are my references.